Okay, so it's 9 o'clock sharp. Let's get started. So hello everyone. Welcome to today's Immune seminar. I think this is quite a special event today. We have a duel. I think this is the very first event on our platform for the last 3 years. But I think I'm sure everyone will enjoy today's seminar. So it's really my great pleasure to welcome today's speaker, Dr. Justin Sonnenburg, and then Dr. Sean Spencer from Stanford to join us for today's lecture. So a very, very brief introduction. Justin currently is a full professor in the Department of Microbiology and Immunology at Stanford School of Medicine. So I think no need to too much detail introductions, since I think a lot of us know Justin's work. So Justin did his undergraduate study in UC Davis, and then further to pursue his PhD training in UC SD. And then afterwards, he decided to pursue his postdoc training in microbiology. So he joined Department of Molecular Biology and Pharmacology in WashU in St. Louis with Jeffrey Gordon. And then from 2008, he started his independent research group as assistant professor in Stanford School of Medicine to 2022 as a just mentioned, full professor. And meanwhile, he also is the director of Center for Human Microbiome Studies in Stanford. So the general goal of Justin's lab is to elucidate the basic machinery which underlie dynamics within the gut microbiome and diverse and implement strategies to prevent and treat disease in humans through gut microbiome. So the general long-term objective is to perform mechanistic studies which set the paradigms of how our microbiota may be incorporated into the emergent version of precision medicine. And then his lab actually investigated the principles which govern the microbiome community function within the gut using extreme-free mice colonized with defined module microbiome community to develop hypotheses which they can pursue within a complex microbiota. So I think with Justin's numerous really remarkable publication record, he has received numerous awards, which including NIH Direct New Innovator Award and the Pioneer Award and the AJ Research Mentor Award. And at the same time, he also co-founded the Novel Biotechnologies, which is a synthetic biology company which engineers gut microbiome to treat diseases. And one of the highlights is he and his wife, which also his colleague, Erica Sonnenburg, are the authors of the book, which I highly recommend everybody to check it out, which is the Good Gut, taking control of your weight, your mood, and your long-term health. So for the second part of our seminar, it's going to take hold by Sean Spencer, who is currently a gastroenterologist and a physician scientist at Stanford. He's currently working with Justin as an instructor, try to discover the role of dietary intake on the gut microbiome and mucosal immune system. And Sean got his MD degree in UPenn, and then further earning his PhD studying nutritional immunology with Yasmin, who's also is in NIH currently. And then after which he moved to Boston for residency training at MGH and complete his gastroenterology training at Stanford. So his general goal is to study mechanisms by which dietary intake influence our microbiome and immune system to better understand and treat the gastrointestinal diseases. So I think it's going to be an exciting lecture. So welcome both of you. And then we are very much looking forward to the talk. Thank you so much, Justin.
Awesome. Sean, thanks so much for the kind introduction, for plugging my book mostly, and for organizing this seminar. I mean, to put it at a time where it's accessible to people around the world to record it, I think it really is in the spirit of kind of what we should be doing more of in science. So thanks for your efforts here. Wonderful to be here and speaking to you. I Sean is in my lab training, but I mostly see him as a colleague. We work closely together on a number of projects, but he also is one of the few bona fide immunologists in our group. And I knew this was an immune talk. And so it's, I hope to be an immunologist one day, but Sean's helping me get there, and his data, I'm sure, will be of great interest to this community. So I'll hand it over to him about halfway through. So my lab at Stanford started about 15 years ago, and I came from Jeffrey Gordon's lab. We really were interested in mechanism, reductionist approaches. We use notobiotic, germ-free mice, and then a bunch of technologies, molecular genetics, to try to get at mechanism. I think I run the lab with my wife Erica, and we quickly became frustrated in this kind of growing roster of really interesting things to study and not knowing how to prioritize these things. And so we started thinking about ways to filter the list of possible things to investigate, and to try to get to things that are disproportionately important. And so we turned to thinking from an evolutionary framework, and then also just kind of from a human filter. We started studying indigenous populations like the Hadza hunter-gatherers in Tanzania, shown here, and a number of other populations in different parts of the world. And then also doing dietary interventions in humans, trying to understand how we can change the microbiome through diet, and how that can ripple through our biology, how changes in our microbiome can ripple through our biology, our immune system, metabolism, and so forth. So I'll talk a little bit about each of these things today. But I wanted to start off just with some general background. This is a cross-section of the colon, and it shows the microbiome in situ. And I really like this image. It was taken by a former graduate student, Kristen Earle, in the lab. And I think, she knew the moment she took this that it just really captured not only the, aesthetic beauty of aspiring stool, but also the some of the really fundamental features of the gut microbiome. So that all the little rods in the top left are the bacteria stained with different fluorescent probes. You can immediately see how dense this community is, and then also how close it is to the host tissue. So that's the blue area in the bottom right. And, it's just separated by, tens of microns by this green mucus layer. So an incredible interface for exchange of biological information. And, we know that there's trillions of microbial cells that are there. The combined genome is staggering. We don't know what most of the genes do in this community. And then, I think we're still in the process of mapping the microbes that are in the gut, and in different populations, different regions of the gut. But we also are really needing to expand understanding what these microbes are doing, how they're impacting host biology. So we've known for a long time microbes can affect our digestion and gut health, but of course, there's just a huge range of our biology that these microbes can control. Virtually no aspect of our biology that goes untouched by our gut microbiome. And while this image really depicts the microbes as being in control, I think a more proper way to think about it that's less microbial centric is just that they're part of this complex integrated system that is our body. But what's special about our gut microbiome is that it's malleable, that it can change. And so because it's connected to so much of our biology, because it is different in each one of us, it means that when we learn the rules and how to manipulate this community, we will be able to change our microbiome in ways that are beneficial for whatever we're going through, whether that's, immunotherapy, fighting an autoimmune disease, trying to conceive, trying to run a marathon faster, whatever it is. So those days are coming. But I also think that this malleability represents an incredible vulnerability of our biology. And it means that an important component of our biology can deteriorate if we're not paying attention to it. And so, fecal transplants for C. difficile colitis have been a crude but vivid demonstration that we can manipulate our own biology in positive ways using the microbiota. And the question is, what are the limits of this now? And of course, there's all sorts of tools, expanding knowledge base, expanding number of trials focused on fecal transplants or administration of microbial therapeutics. I think one of the amazing things that runs through virtually all of these studies, and certainly has run through our research program from the beginning, is the importance of diet in dictating the composition and function of the gut microbiome. And so I wanted to just lay out in a very simple graphic here, the schematic of kind of the logic of how we think about diet impacting our gut microbes. So, we have microbes, of course, all over our digestive tract. Most of them reside in the distal part in our colon. And because there's so much microbial mass there towards the end of the line, it means that they are not getting the nutrients that we are consuming the most of. So when we eat something like cake, but certainly most of the other food we eat in the industrialized world that's filled with protein and fat and starch and sugar, those foods come in, they get digested and absorbed in the small intestine, and that leaves our microbes without much to eat. And the kind of flip side of this is if you're eating a complex plant-based diet with tons of dietary fiber, those complex carbohydrates are not digestible in our upper GI tract, and then can make it to the distal gut where our microbes can ferment them and turn them into short-chain fatty acids. So this kind of longitudinal vision of the or schematic of the microbiome really lays out why we think dietary fiber, and particularly one component of dietary fiber, a portion that we call microbiota-accessible carbohydrates, or MACs, the part of dietary fiber that can be fermented by the gut microbiome, why that's disproportionately important to our gut microbes. And we certainly know there's been work out of our group and other groups like Eric Martin's group that have shown that if you're not eating a lot of dietary fiber, your gut microbes will actually start eating you. So that mucus layer is made of carbohydrates as well, and it serves as a backup food source for microbes that are need consistent endogenous source of energy and calories. So in an experiment a while back, we really wanted to look at the role of dietary fiber in impacting our gut microbes and impacting the diversity of our microbes. So we actually took germ-free mice, gave them a human microbiota, gave them a high MAC or high fiber diet, let the microbiome equilibrate, and then shifted half the group onto a low MAC diet. So this would be representative of a Western diet. We knew that the diversity of the microbiome would crash during this time. And then the question was, if we brought back a high fiber diet, would the microbiome come back? Would the diversity rebuild? So this is essentially asking the question, is it too late for us, or can we just start eating a healthy diet and hope to resurrect our gut microbiome? And so we were surprised to see that actually a lot of the diversity came back in this experiment after it had been depleted. So the high MAC diet was good at bringing back microbes that were probably just suppressed to low levels. But we thought there was probably something more going on here. And so we ran the same experiment, but over 4 generations. So 4 generations of mice on either a high MAC, again, high fiber, or a low MAC diet. And so on the high MAC diet, what we see here is the 200 most abundant species in the different columns, and their abundance over the 4 generations sampled in 5 mice at 2 time points. So on the right here, you can see microbes that are rock stable as they're passed from generation to generation on the high MAC diet. Other microbes are more variable through the 4 generations, and then a few even go extinct over the course of these 4 generations. And so that's the high fiber diet. What happens on the Western low fiber diet? And so that's shown here. You can see just massive extinctions that compound over the course of the generations. So elimination of dietary fiber depletes the gut microbiome irreversibly. And notably, at the end of the experiment, we bring back a high fiber diet to see if the microbes will come back, and they don't. So once these microbes are lost, they're not available, they're out of the system, and the microbiome has been depleted to about 1/3 of its original state, and it's locked in that state without access to these microbes that have gone extinct. So that's mouse data. At the same time, we were performing human studies, and we performed a study in the Hadza hunter-gatherers, and that mainly focused on seasonality of the Hadza gut microbiome, the fact that species go away and come back in a seasonal way as foods fluctuate. But one of the more important figures out of that study, Sam Smith's a former graduate student, brought together a bunch of data from other populations, microbiomes that was available at the time, and put that onto one principal coordinate plot. So all of these bars represent gut microbiome composition determined by 16S sequencing of the populations indicated on the left, and then arrayed on this principal coordinate plot. So where they are on, where the populations are on that axis actually gives you an idea of the other populations that the microbiomes are similar to. And this tells us a really kind of stark finding that the non-industrialized populations have a very different microbiome than industrialized populations, something that has become kind of more common knowledge now. But at the time, it was a really, really striking pattern. And what was more impressive was that this isn't just loss of diversity. If we array bacterial families, relative abundance across that same axis, these are families that explain the variation in the data. We can see there are families like spirochetes, succinivibrio, and prevotella that are all very abundant in non-industrialized populations and are depleted in industrialized populations. And the reverse pattern is true for Verruca microbia. So Akkermansia mucinifila, a mucin-loving bacteria that is very good at degrading mucus, becomes more abundant in industrialized populations. We can also show blooms of Akkermansia in mice when we deplete dietary fiber, but here we're seeing it on a global scale. Populations that eat low fiber diets have increased levels of mucin degradation. And so we performed studies along lifestyle gradients in Nepal. I don't have time to go through that, but Ashish Jha, who led that study, now faculty at NYU Abu Dhabi, proposed this model where we can see that there are different patterns of microbiome loss and gain across the lifestyle gradient. So foraging lifestyles, agricultural and industrial, a lifestyle gradient that also represents our evolutionary history, because we all were foragers. If you go greater than 10,000 years ago, around 10,000 years ago, we started farming, and then gradually have become more industrialized over time. And so these taxa that go missing, we call the vanished taxa, volatile and associated negatively with industrialized societies of humans. And then the ones that appear, we call the blossom taxa. Blooms are selected in societies of urbanization and modernization. And so recently we just published this paper in Cell with a broader characterization of the Hadza hunter-gatherer gut microbiome, but put it in the context of 1,800 different global metagenomic data sets from the microbiomes of populations around the world. And in this heat map, it's actually just a binary plot. We're looking at the presence and absence at a given threshold of the 500 approximately most prevalent species within any of the groups that we're surveying. So Hadza hunter-gatherers, transitional populations, and then industrial populations. So we see the exact patterns that Ashish predicted in that paper. We see groups that become more abundant in the industrialized populations. These are the blossom taxa. We see taxa that go extinct or become very rare at that same boundary, and then others that are more specific to the hunter-gatherer lifestyle. And so now we have a really refined idea of species that are being lost and gained. And this leads to the question of, how does this fit into the context of an evolutionary framework? Because what we have here are all modern humans living on present day Earth with different lifestyles, microbiomes. And so there was an incredibly important study out of Alex Kostik's lab at Harvard a couple years ago, where they sequenced paleofeces. So these are fecal samples that have been fossilized, humans that lived 1,000 to 2,000 years ago in North America. And they determined by sequencing the composition of those paleofeces and then overlaid that composition onto a principal coordinate plot with other sequence samples in the database from industrialized and non-industrialized populations. And so we see this really clear pattern here that the paleofeces overlays with the non-industrialized microbiome compositions. and that the industrialized populations have different microbiome composition. So this is telling us that these non-industrialized populations are more representative of an ancestral state, as you might imagine from their lifestyle, and that the industrialized microbiome represents a recently derived state that departs from the microbiome that, or features of the microbiome that existed in humans for, tens of thousands, probably hundreds of thousands of years, and probably played a role in shaping our human genome. So would our health benefit from bringing back lost species or functions? This is an important question, one that we don't have the answer to. I think it's clear that we've lost some pathogens that we'd rather not bring back, but we've lost, dozens to hundreds of species in our gut microbiome. I think it's probably and without any foresight or rational guidance of the microbiome, I think it's probably likely that we've lost species that are important in interacting with our human biology. And the key now is to figure out what things would be beneficial if they were, present given the modern conditions. And this is, of course, a big question, and also brings to light issues of protecting indigenous communities and making sure that bioprospecting isn't occurring. All of our research with these indigenous populations is strictly for research purposes, but I think it does suggest that there may be pieces missing to our industrialized microbiome that may be important for our health. So we know the microbiome is important in regulating immunity, shown very vividly in many animal models. We know that our microbial identity has changed in the industrialized world. We also know that many chronic inflammatory diseases are on the rise. I've just shown some examples here, but the list, of course, is much longer. And it brings up the question of whether, in having a new or kind of foreign microbiome that exists inside of us, could we have potentially a misregulated immune system that's driving us towards different inflammatory diseases? And what disease you develop will depend upon aspects of your human genome. We can find those out by GWAS, but what really is setting up the big problem is the change in our gut microbiome. So a major question we tried to address is, can we repair our microbiota and improve our health? And so we several years ago started the Center for Human Microbiome Studies with a great group of partners at Stanford. And the idea here was basically, I learned to do science in this kind of forward translational method, molecular mechanism through animal models and then through humans. And this is basically asking the question of, if we develop humans as an experimental model, can we run this in reverse? Can we reverse translate things that appear to be associated, features of the microbiome and aspects of the immune system, and reverse translate that into animal models so that we know what we're studying is relevant to human biology. And so we started this center with the idea that we could do kind of longitudinal dietary interventions, monitoring the microbiome, and then monitoring a bunch of immune features with wonderful immune profiling that Mark Davis and Holden Maker have set up through the Human Immune Monitoring Center at Stanford. And so all of these dietary interventions we do with Christopher Gardner's group. They're really the wonderful collaborators that get free-living humans to eat differently. And so I just wanted to tell you quickly about one of our studies. This was actually kind of the flagship study that we published in 2021. We call it FIFO for fermented fiber food. It's basically, a two-arm study where we feed one group of people a high fiber diet, legumes, whole grains, vegetables, fruits, and another group high fermented foods. So foods containing live microbes, kombucha, kefir, yogurt, kimchi, sauerkraut. And over the course of this, what in total is a 17-week protocol, but 6 weeks of maintenance phase for the intervention, we ask whether we can change the microbiome and whether there are any changes in the immune system that go along with that. And so a wonderful group of trainees has driven this forward. Maddie, Hannah, and Gabby, they've done a phenomenal collaborative job with a big group of people to bring all this data together. And it tells a pretty interesting story, and perhaps one that we didn't fully anticipate. So again, the high fiber diet, just plant-based, high fiber, high fermented foods are all of these foods that you buy in the refrigerated section that have live microbes. this is what we achieved, or what Christopher Gardner's group achieved in terms of getting people to eat differently. Fiber intake went from about 20 grams per day up to over 40 grams per day on average in this cohort of healthy adults. So they were over doubling their dietary fiber intake. And then the fermented food group went from close to 0 fermented foods up to over 5 servings of fermented foods during maintenance. And then we also have this choice period, which is like a washout period, but Christopher rightly refuses to force people to go back to a crappy diet at the end of our studies. So he lets them choose what they want to do. So they enjoy these diets. They stay on them to some degree, even after the maintenance phase is over. Okay, so we got people to eat differently. What happens to their gut microbiome? So this is microbiome diversity in terms of number of ASVs from baseline through the study. High fiber didn't move the microbiome across the cohort, very flat, but the fermented foods increased microbiome diversity significantly at the end of the study. So this was very interesting to us. I won't have time to go into the details, but we now know that it's not the microbes in the fermented foods that are actually making up this new diversity. They only make up about 5% of the new species. It's mostly resident microbes that either come in from the outside or bloom from low levels. And we're trying to figure that out now. So why didn't the fiber impact the gut microbiome in a bigger way? Well, we think that our microbiome has lost a lot of the fiber degraders. And so over the course of this short dietary intervention, the fiber may just shoot through us untransformed because we don't have the right microbes there. And indeed, when we do analysis of stool carbohydrate, we see a lot of the monosaccharide markers of hemicellulose that should be, and pectins that should be fermentable. So it indicates that indeed the diet is overwhelming the fermentation capacity of the industrialized gut. And this corresponds nicely with beautiful work that's been done at University of Minnesota. Dan Knight's group showed that as immigrants come to the United States, their microbiome changes within months, and then that change becomes more severe over time and over generations. But one of the big changes is loss of fiber degrading strains in the gut microbiome. So this loss and getting back to our multi-generational experiment, when you lose microbes, it's very hard to reacquire them. Especially in a very sanitized world that we live in. One potential note of hope is that in that multi-generational experiment where we depleted the microbiome with a low fiber diet, if we use the high fiber diet mice as the donors and did an FMT, so high MAC diet plus microbes, we actually can restore all that diversity that was lost. And that comes back very rapidly, and it's maintained over time as long as that diet is there. So it may be that diet isn't enough. We need diet plus the bugs to reconstitute the diversity. So as I mentioned, the diversity goes up in the fermented food fed people. What happens to their immune system? So each one of these is a different O-link measured cytokine or chemokine over the course of the intervention in this cohort of 18 healthy adults. We see many markers come down over the course of the intervention as their microbiome diversity is going up and they're eating fiber. And that's just a small snippet of a larger signature. Over 24 markers measured by O-link and then CyTOF show decreases in inflammation over the course of this intervention. So an indication that we can change our immune system by eating fermented foods and potentially changing the gut microbiome diversity. And I'm going to stop sharing there and I'm going to hand it over to Sean.
Great. Thank you for that, Justin. Always great to see. I will now be switching gears, and Justin set that up very nicely to suggest that the fermented food intervention in humans was interacting potentially with both the microbiome and the immune system dynamically. And this highlights really nicely kind of what I ended up in my PhD studies with Yasmin publishing an opinion piece at the time, suggesting that diet, immune, and microbiome interactions were intertwined and really hard to disentangle. And really it left the key question of how to identify mechanisms of interactions to understand disease and develop therapeutics. And I think I was very much motivated to come to Justin's lab for my postdoctoral studies based on the model that he was laying out. And actually, when I first joined his lab, we put forth this perspective, really laying out a proposal for this model of longitudinal studies in humans with reverse translational mechanistic studies in mice, as Justin described very nicely. And I'd like to give you a bit of an example of that today, what we've done with the fermented food study in humans. And really, I want to also say that this is not a linear process. It's very much dynamic and cyclical. And our hope is that by briefly dwelling in a reverse translational space, that we can better inform a next clinical trial that we could perform in humans. So fermented foods were nicely described by Justin. I just wanted to give a bit more background of why fermented foods are an interesting thing for us to study. They're ancient and they're widely consumed prior to industrialization. So this may be one of those factors that is driving a non-industrialized microbiome state. We have evidence that even before humans evolved, that our ancestral fruit foraging apes evolved enzyme, alcohol dehydrogenase, proposed to have been developed so that they could consume fermented foods with greater access to nutritional capacity. And finally, fermented foods are considered to confer health benefits, but not well tested in interventional clinical trials or mechanistic animal models. And so this is the cytokine data that Justin showed, and it really suggests that fermented foods reduce inflammation, but the mechanism remains unclear. Are these purely microbiota-directed effects, or are they host-directed effects? Likely both. And what components of fermented foods are mediating this effect? So to start out with, I just asked a couple of questions. I first sought to define what fermented foods were chemically and microbiologically, and really to ask how fermented foods affect the host and the microbiota. And today I'll be mainly discussing how fermented foods affect the host. And the main immune cell that I'll be focusing on is T regulatory cells. We've done a broad immune survey, but I think the effect of fermented foods on T regulatory cell homeostasis in the gut nicely highlights our findings. So first of all, we took advantage of a new metabolomics pipeline that was developed in the Sonnenburg lab to really probe the chemical content of fermented foods from a microbial metabolite perspective. And this was an LC-MS pipeline. And what we did is we took the fermented foods that were consumed by the participants, and we ran them through this pipeline. And what you'll see is an unsupervised hierarchical clustering of the top 90 metabolites found in at least one sample. And what you can tell is that the fermented foods cluster by type. So on the bottom, you have your vegetable ferments, then you have your yogurts, kind of the kombuchas group with a juice control, this turmeric shot, and then you have goat kefir and vegan milk yogurt at the top. And you see in these red bars of the high concentration metabolites, these are really what we're interested in going forward for potential mechanistic studies. Also worth highlighting that these are the mediators of flavor. And we know evolutionarily that things that had flavor are more likely to have biological impact and potential health impact. So there is likely a coevolution between things that we find flavorful and those that have biologic impact in the gut. And because we had this nice data set, we really wanted to ask, can we leverage our data to generate a testable hypothesis? So we said that the fermented foods increase microbiome diversity. So we performed a linear regression, and this is in the original publication, basically looking at types of fermented foods. And the types of fermented foods that had the greatest association with microbiome diversity in general were yogurt and this vegetable brine drink was essentially the juice from fermented vegetables. That's sauerkraut brine. And I told you we had done the LCMS pipeline, but we also wanted to complement this with the GCMS pipeline to look at organic acids, which the short-chain fatty acids belong to, which are known to have potent interactions with the immune system. And what you can see here is that the yogurt and the gut shot have very high amounts of lactate, which is not surprising because they're lactic acid fermentation. However, the kombucha, even though on the label they say they add lactate, quickly shunts to acetate, and it's mostly acetate. And the yeast-based koji ferments, miso included, do not have high levels of any of the organic acids that we looked at. And really the main point here is that these levels are quite striking. So the level of lactate in this fermented vegetable brine is about 100 millimolar, and in the trial, patients were consuming up to a bottle of this per day. So, you know, upwards of 10 to 15 grams of lactate that's being orally ingested per day. And we next wanted to investigate how fermented foods affect the host. And I think this is one thing that has limited studies of fermented food to date, is that they're complex. They have macronutrients. There are prebiotic fibers within them. There are probiotics, live microbes, and there are metabolites. And really the key struggle for us was how to really ask a mechanistic question with this complexity. And so we landed on the this fermented vegetable brine because it had several advantages. It didn't have significant macronutrient content. It was not a significant source of prebiotics, and it was only a milieu of live microbes and their microbial metabolites. It seemed like an ideal place to start. And so for these studies, what I did is I took the commercial product, I administered the whole brine to mice, or I sterile filtered the brine to deliver mainly microbial metabolites in the absence of live microbes through a 0.22 micron filter. And then because lactate is the dominant metabolite of lactic acid fermentation, really starting from the bottom up of what is the effect of lactate itself. We utilized for this study X germ-free mice. So we colonized with healthy human stool 7 days before administering by gavage, either a control, which was saline and acid control, and fermented vegetable brine intact. We saw a couple of striking things. The first was that we see an expansion of T regulatory cells, primarily in the small intestine, pretty dramatic expansion that's not seen in the large intestine. This was surprising to us at first, but in retrospect, perhaps not that surprising, in that the primary exposure site for ingested food is the small intestine. You know, these are microbial metabolites that really bathe the small intestine, and this may be more of a pharmacologic issue. And again, not apparent in the large intestine. And we next wanted to use the different brines that we had generated. And before I move on to show you the other results, I just wanted to highlight describe more the expansion of Tregs. So this is an increase in Tregs by percentage, as well as by total numbers. We see a slightly greater expansion of ROR gamma T Tregs, but this does seem to be a broad, non-selective expansion of Tregs that crosses all Treg subsets, suggesting that we're broadly affecting Treg homeostasis rather than a single subset. The expansion of Tregs does seem to be fairly local, really predominant in the small intestinal lamina propria, present but less striking in the mesenteric lymph node, and again, present but intermittently significant in the spleen, suggesting that this may be a local effect in the small intestine. And we've also done extensive studies in conventional mice and in mice with synthetic communities, and we do see this on a wide range of microbiota backgrounds. Then for one of the more exciting experiments we did was we really wanted to probe the effect of metabolites themselves. So this is asking, what is the role of metabolites within fermented foods on the immune system? I think if you were to ask most people, why fermented foods are beneficial, you would hear either the prebiotic capacity, the fiber within it, or the probiotics. I think these are what most people view as the beneficial component of fermented foods. But much to our surprise, when we administered the metabolites alone, we saw a strikingly similar effect on T regulatory cell homeostasis, suggesting that at least this phenotype in the small intestine can be recapitulated by metabolites alone, really suggesting that metabolites in fermented foods are an important bioactive component as well. And then lastly, when we used lactate alone, we were also surprised that lactate could largely recapitulate this phenotype in this setting. And then next, we wanted to do a head-to-head. As I showed you earlier, the primary metabolite in kombucha is acetate, whereas in vegetable lactic acid fermentation is lactate. So then we did a lactate acetate head-to-head of just a single metabolite. What we saw is that lactate, again, increased Tregs, but orally delivered acetate does not alter Treg homeostasis in the small intestine. This is consistent with a large amount of the short-chain fatty acid literature. It's because acetate is avidly absorbed in the duodenum and probably is not bioavailable distally. And then just in the last 10 minutes, I wanted to discuss a little bit of where we're going from here and really thinking about this concept of how microbes versus their metabolites interact with the immune system. I think we've known for a long time that quote unquote probiotics, those bacteria with beneficial health properties, have ligands and metabolites with reduced inflammatory potential. Whereas pathobionts and frank pathogens typically thrive in settings of inflammation and have ligands and metabolites with increased inflammatory potential to create a niche that they can thrive in. And we really wanted to ask, kind of inspired by our initial findings, can we leverage microbial metabolites as a lever to control microbe-immune interactions in the gut? And really, to put it a different way, can we alter GI immune responses using orally delivered metabolites? Basically, the way that the immune system perceives bacteria is through the ligands and proteins that it sees. So basically, MAMPs and proteins that the T cell can respond to, but also the metabolites in the milieu condition the antigen-presenting cells and the T cells themselves with what flavor of response and which polarization they'll respond to. So we did two complementary experiments. We wanted to do a gain of regulatory function, and we wanted to have a loss of regulatory function. So essentially what we did is we colonized germ-free mice, monocolonized with them with E. coli, and then we gave E. coli plus only the fermented food metabolites. Conversely, we took a probiotic, L. plantarum, colonized germ-free mice, and then we colonized the same strain that is knocked out for its capacity to make lactate. So it's a lactate dehydrogenase knockout and has no lactate production. And then our readout again was immune regulation as demonstrated by Treg homeostasis. So essentially, we wanted to make E. coli a probiotic and make L. plantarum less probiotic through toggling its metabolic function. And so this is a demonstration. We just used typical lab E. coli MG1655. And then you can see not really a big colonization difference, perhaps a single outlier in the distal, but otherwise the administration of brine doesn't significantly alter E. coli homeostasis along the entire GI tract. And as consistent with prior literature, monocolonizing mice with E. coli MG1655 does not really engender that much of a regulatory response. It doesn't engender that much of a T cell response in general. And much to our surprise, when we administered the sterile fermented vegetable brine, you see this really dramatic expansion of T regulatory cells in the intestine. And because this was a monocolonized state, you did see this in the large intestine as well. Most likely because in the monocolonized state, if your immune system is only responding to E. coli and with limited metabolic diversity, otherwise you can affect the large intestine as well. We then did the inverse experiment of making a probiotic less probiotic by administering the L. plantarum knockout. And you can see maybe a bit of a colonization defect in the proximal small intestine, but not significant. Most importantly, when we look at lactate production in the mouse, you see almost no lactate production. And what we saw is, and this is at homeostasis in the small intestine, is that you get significantly decreased T regulatory cells in the small intestine when a probiotic is not able to make lactate. And what's also interesting is it looks like there's a hint that you start to polarize more towards a pro-inflammatory Th1 state in the absence of lactate, suggesting that that the LDH gene function within probiotics is very important for promoting regulatory T cell responses and potentially for restraining inflammatory responses. So where do we go from here? This is how we're thinking about unraveling these complex interactions just for a single metabolite, for lactate. And as I demonstrated, there's hundreds or thousands of metabolites within fermented food. But what we want to understand is, how does lactate affect the microbiota directly? Can constituents of the microbiota use lactate? I didn't have time to show the data today on the microbiome, but we do have data suggesting that bugs within the microbiome can utilize lactate directly. And this may affect homeostasis in trans via the microbiome. We're very keen on understanding mechanisms of direct metabolite sensing. There are a couple of mechanisms by which lactate can affect homeostasis. It's very well appreciated that most cells of the human body can utilize lactate as a fuel via import into the cell via MCT1. We have collaborators who have a lineage-restricted MCT1 deletion on Tregs. Lactate can also be sensed by the myeloid compartment and antigen-presenting cells via two G protein coupled receptors, GPR81 and GPR31. And we have lineage-specific GPCR deletion of 81, and we have a global knockout of GPR31. And to better understand the role of lactate in trans via the microbiome and its effect on the immune system, we're building synthetic communities from the ground up in notobiotic mice to really understand what are the requirements for different microbiome constituents for having an immune effect. But we do know from the E. coli experiment that really just a single bug is sufficient to have this effect. And then really, so I mentioned one metabolites, and I think a key question that we're grappling with right now is what other metabolites are present, and what is the best approach to prioritize these metabolites for in vivo testing. And this is active work that's going on in the lab of really trying to decode all of these interesting metabolites in a variety of fermented foods. But as a physician scientist, I think the thing that excites me most about fermented foods is really thinking about how we can return this mechanistic data to clinical trials and asking the questions, can we make a medical grade fermented food to treat inflammatory disease and promote a healthy microbiota? I think something that limits our ability to use probiotics and fermented foods in states of inflammation is that there are case reports of in settings of impaired barrier function, particularly in ICU setting, or in inflammatory bowel disease, of probiotics causing sepsis or endocarditis. And it is interesting to think about a medical grade fermented food that maybe consist of metabolites alone in the absence of live microbes that may have a dramatically improved safety profile in inflammatory settings. And then second, can we tailor the metabolite milieu to confer specific immune functionality? And I think, as we look at this diversity, I think undoubtedly, as we look into specific metabolites, I think some may be pro-inflammatory, some may be anti-inflammatory. And it might be interesting to think in the setting of checkpoint therapy for cancer, you may want to deliver some of these more isolated pro-inflammatory metabolites to increase immune responses in the gut and to potentially not promote tolerance. However, in settings of food allergy or immune homeostasis and just for gut health in general, you may want a regulatory milieu. So I do want to to demonstrate that we are thinking about this in a complex way. And I don't think that all fermented foods will be purely regulatory. And I think it will be important. These mechanistic mouse studies will be critically important for understanding how we can tailor basically bacterial metabolite therapies for specific precision medicine approaches. And with that, I just want to end, and I think Justin can come on and say thanks, but I just want to specifically thank Justin for having me in the lab and for everyone who's helped me with this project.
Great. And